happy to be here and to present something I think very specialized that I had in my career a lot of questions of bioassays tests, and not a lot of people are aware that you can do a lot in JMP. It's maybe something hidden in some of the menus, but everything that you are using in bioassays, and I will introduce you a little bit more about the terms, everything you can do very, very easy in JMP. Today we will talk, or I will talk about analyzing the potency and stability of bioassays. So actually I'm a molecular biologist, and in my previous career before joining JMP, I had a lot of those types of analysis, and it was a little bit cumbersome to do everything manually. A little introduction before I'm going to the demonstration is a little bit about bioassays. So what are bioassays? So bioassays are tests in biology, in cells, in tissue, in vivo or in vitro. And the main idea is to find the potency of substances. So how do they react with ligands? Are they the same with standard references? And I've put here some terms that I found in internet. A bioassay is an analytical method to determine concentration or potency of a substance by its effect on living cells or tissues. So you can estimate the potency of agents by observing their effect on living animals, so in vivo, and also in tissues or cells in vitro. And the main questions that I think the authorities are asking if you want to submit something is, are your tests or are your curve parallel? So you might do some parallelism tests. So the main question that you might answer is, do the curves have similar slopes? And this is something very, very important. This is, I think, the first thing that you need to do is to do a parallelism test. Also, relative potency. So does my sample produce this desired response compared to a reference sample when tested under the same conditions? So potency would tell you if one enzyme or other product is more potent or more active than others. Equivalence test is also very important, is to estimate if your curves are similar. So depending on the shape of the curve or on the model that you apply. Here, for example, if you have a 4P or 4 parameter model, you might test if the asymptotes of the lower or the higher are the same, if the growth rate is the same or different, and also the inflection point. So those 3 different tests can be done easily in one platform that I will show you afterwards. Things that are also very interesting is, for example, to make inverse predictions. So, for example, actually, let's say you have a curve on the x-axis, you have the concentration, and then on the y-axis, you have the toxicity or whatever, and you would like to predict at which dose, or what will be the dose for a specific response. So that would be the inverse prediction. And this is very related to what a lot of people are using is the EC50 or IC50. So it's actually also an inverse prediction. And this is very helpful to find out what is the concentration or the dose effective in producing 50% of the maximal response. So for example, 50% of toxicity, what will be the dosage for that? So this is the EC50. And this is also very related to the relative potency. And what I was also looking, in other types of estimations like Vmax and the Km estimation. So this is the Michaelis-Menten constant. So, and this is also very helpful to find out what is the affinity of the enzyme to the substrate. So all those different terms, I will show you how you can apply that using JMP. Before doing that, just wanted to tell you that I've published a blog that will actually apply everything that I will show you. What I will show you today is actually documented. First of all, where do you find all those different things? So you might apply, those response types of analysis. So those response are obviously not linear. It could be linear, but I would say 90% of the cases that are non-linear models. And of course, you will not be able to use linear models to answer those questions. Therefore, in JMP, we have a platform in, as I said, in JMP and also in JMP Pro. It's called the Fit Curve platform. Where do you find it? It's under the Analyze menu in Specialized Modeling and Fit Curve. So this is a really, really nice platform because you have a lots of models that are already built in, right? So you don't need to type manually the model. That would be maybe for this nonlinear model. So it's use custom functions. So you can also use this one, but it's a little bit more, I would say, not very practical. I really like the Fit Curve because it allows you to use a lot of different models that are already built in, right? And you can find them, maybe it's a little bit small, but you will find here a lot of different curve models that are already built in for polynomials, for sigmoid curves. So that would be, I think, the most model that I will use today. Also for logistic curves, so sigmoid curves, other sigmoid curves, exponential growth and decay, peak models, pharmacokinetic models, and also Michaelis-Menten model that I will show you at the very, very late stage of the presentation. So this platform is very, very useful, in situation where you have well-known nonlinear equations that describes the relationship between the variables, and especially in the field of physical and biological science. As I said, I would say the sigmoid models are very, very useful for those response types of analysis. Michaelis-Menten are very also useful for enzyme reactions. But you have also peak models, as I said, exponential growth, simple linear regressions, so you can also use that here, and also pharmacokinetic models. You will find in the documentation, again, I have a link here that is actually pointing me to all the different models. So everything is very, very well documented in JMP. I will not go through all of that because I think it's a little bit boring, but you will find the formula, how the models are, all the different parameters, and so forth and so forth. All right, so this was a little introduction on the Fit Curve platform. Of course, I will use this Fit Curve platform all the time in my presentation. So I think you will be very familiar once I will be at the very late stage of my presentation. So the parallelism test, as I said in my introduction, I would say is the first type of test that needs to be done in order to find out if the curve have the same slope. So for example, you would see if you have different formulations that you want to test and then to compare, the first thing that you need to check is if the curves behave similarly or in parallel. So that would say that at every time the distance between the two curves are the same. So if you extrapolate that on the log dose, so the dosage would be always the same. Of course, will be different, but the difference between the two are always the same, right? If that's not the case, right? So that would be something, a situation like that, if you have two curves and in any time you have a delta that is different. Yeah, so you need to test if you are in this situation or in this situation. And of course, this parallelism test is, so the shift of the two curves it's not important, it's only the differences between the shift are that are important. So in JMP, I just wanted to show you how you can do that. But before doing that, I just wanted also to introduce you a little bit about the fit curve platform. And I have a data. It's actually data from the sample data library. It's called Bioassay. And this data has a different formulation. And let me show you that here. So you have 4 different formulations. You have a standard, you have a test A, test B, and test C. And all those tests has been measured for the toxicity based on the concentration or the log concentration. And we would like now to find out which type of curve fit best to this data set. What I'm usually doing is first to check that with the graph builder, so to see if the data is a good quality. And the graph builder is a very nice platform. I suppose that a lot of people who are starting using JMP actually start using the graph builder because they see really the power of doing a graph very, very easy and very fast. So I will put the log concentration on the x-axis and then the toxicity on the y-axis. So we can already see this particular curve, but as we have 4 different formulations, I need to use this formulation. I can use that as a group X. That might be useful, but in my case, I would like now to compare those different curves, right? So if they are different or not. So what I would need to use is actually the overlay. So now we can see here on the overlay the different curves of these 4 different formulations. And we can already see that there's one curve that behaves differently, and this is test B. All the rest are quite similar to the standard. So test A and test C are very, very, very similar. You will find that out also using the different tests, that the test B is behaving different as all the others. Okay, so I think the data looks good. So I will show you that using the fit curve. So the fit curve is under the Analyze menu, Specialized Modeling, and Fit Curve. So here's the entry point. So you might have different data formats. So in my case, I have a stack data. So everything is one column, right? But you could have also rows as columns and rows as functions and columns as functions. So depending on how the data is formatted, you can use one or the other. So now I have to populate my different fields. So the toxicity will be actually the response, will be my Y. The log concentration will be my X-axis, the regressor. And my formulation will be the group variable. I might have also supplementary data. So supplementary data would be, for example, something that is not really important in the curve by itself, but could be used to find out what are the effects of variables on the curve shape, right? But I will not go into that today, but just that you know that it is also possible. I will click on OK. And now we will see what I've already shown you in the graph builder. Here are my different curves. So you can see all the curves in one graph and then the different curves in the 4 different graphs here. So now there is no model that has been applied. So the obvious thing is now to find out which model will fit best the data. So in the little red triangle, you will find all the different models that I've introduced you. You have polynomials, you have sigmoid curves. Here you can see that you have logistic curves and probit curves, the exponential growth, the peak models, the pharmacokinetic models, and so forth. So in our case, as you have already seen, it's using actually more or less a sigmoid curve. And let's find out what would be the best curve. So I would first try the logistic curve. And it seems to be a 4-parameter because you have here the lower asymptote, the upper asymptote, and then you have the growth rate and also the inflection point. So you will have 4 parameters. So in my case, I will use the fit logistic 4P for 4 parameters. And then now the model has been applied to the data. So you can see here different things. You can see the estimation of the different grouping of variable like the growth rate, the inflection point, the lower and the upper asymptote. And we can already see an obvious thing is that the test B, you can see the inflection point is smaller than all the rest. Again, you know, reflecting here that the test B might be more potent. So the curve is much faster, or the enzyme is much faster in the reaction or in the toxicity, right? So if I had used another model, let's test maybe another model, and then to find out if the model fits best or worse, I would use, for example, in the sigmoid curve, the probit curves, right? And I will use the fit probit 4-parameter. And you can see it's very, 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 very similar to the logistic one. Might be a little bit different. And if you want now to find out what are the differences between the different models, you will find on the top this model comparison. And you can see if you had a lot of models, so you would find out the one that fits best. And apparently the logistic 4P fits best because we can see the AIC. The AIC is an indicator of how good the model fits the data. Rule of thumb is the AIC that's always the smallest is the best model, right? So here we are in minus values. So this value is smaller than this one, but not very, very much, right? And we had already seen that in visualization that the models are very similar. But for now, I will use the logistic 4P. So what I can do is actually to remove the other fit. So let's go to the red triangle, and then we can remove the fit. Okay, so what else should I say for this one? I think that's fine. Yeah, maybe what we can also do is to see the plot actual by predicted, yeah, to see if the model fits a linear regression. We can do that, and then now we can see that the model is fairly good. Yeah, you can see that all the points, if you have a model that is really perfect, all the points would match this 45-degree diagonal. And of course, we have a little bit noise, but it's really okay. And we can see that the overall model and then the model for the 4 different formulations. So the model is good, so we can go forward and then to do this parallelism test. Yeah, you will find in not all the models, because I've checked that before, not in all the models you will find those particular tests because some tests are not applicable. But in the logistic 4P, all the tests that I've introduced you are in the model. So you can see the first one is the test, the parallelism test, right? So it says the test whether the fit curves have a similar shape across the groups. So now I have run the parallelism test, and then we can see a lot of different values. So if you are not a statistician, so don't be afraid. There are very particular things that you have to check. Yeah, and this is actually the p-value. So if I'm mousing over this value, it says that this value is not significant at all. So it says that it's a high p-value. So if you have a high p-value, then you can be confident that your curves are parallel. Yeah. And you can also see that here that all the distance between the curves between test B at least and all the other tests are very, very similar to each other. So here it says that we are in agreement that our different curves are the same or have the same slope. So if that would not be the case, then the p-value would be different, would be very small and would be red or orange, right? And I will show you here with another data. I will hide this for one. I have another data with curves that are not parallel. I can also show you that in the graph builder. So using the log concentration as my X, and then the toxicity that are not parallel, and then using the formulation as an overlay, you can see that the standard here in my case in red behave quite differently as all the rest. Yeah, so that might be something that we need to test if our curves follows or not this parallelism test. Let's show that using the fit curve. So I will use the toxicity non-parallel now for my response. I will use the log concentration as my regressor, and then the formulation as the grouping variable. And then let's do the same. I will click OK. And you can already see again here in this small graph, maybe I can increase that a little bit, that the standard, so the red curve is fairly different as all the rest. I can use, again, the same type of model. I will use the sigmoid curve, the logistic curve, and the fit logistic 4P, right? And we can also see here that the standard curve is different, right? But now we have to test it. We have to validate it. And how to validate it is using the parallelism test. Test parallelism. And now we can see here the p-value is different as the first example that I showed you, right? You can see that the p-value is small. And if I'm mousing over, it tells me also that this value is small enough to indicate a very convincing significance, meaning that the curves, well, altogether are not parallel. The nice thing that you can do also is to find out which curve is not parallel, because here it doesn't tell you really which curve is not parallel. So it's using, for example, the local data filter. I will maybe close more or less all my different windows in order to have, oh, I need this one, of course, I need here the p-value. And I will use here the different formulation in my filter, right? And now I could say, well, I want to see, well, I know that the standard does not behave the same, but what is with the A, B, and C test? So I could use only the A, B, and C test. And now we can see that the p-value is again not significant, meaning that A, B, and C are parallel. That will not be the case if I'm using now, again, the standard. And you can see now the p-value is small again, indicating that the standard is really the one that is behaving differently as all the others. So this was actually the parallelism test, so very, I would say, straightforward, yeah, to find out if your curves are parallel or not. So this is really the first thing to do, because if they are not parallel, well, you will not be able to do any other tests like the equivalence, but because they are not equivalent, right? Or you will not be able to do a potency test, because potency is really related on the on the shape of the curve, of course. So let's go back to my journal file. And let's going on with the relative potency.